Hello students. Now let's see the next enteric pathogen, that is Shigella. So our objectives are classification, biochemical characters, antigenic structure, viability characteristics of Shigella. Now Shigella. Shigella, it is a genus of bacteria that is gram-negative, facultative, anaerobic, non-spore forming, non-motile, rod-shaped and genetically closely related to E. coli. It was first discovered by Kiyoshi Shiga in 1897. Okay. Shigella infection, which is also called as shigellosis, is an intestinal infection caused by a family of bacteria known as Shigella. Understood? The main sign of Shigella infection is diarrhea, which often is bloody. Okay. Shigella is found in the intestinal tract of infected people and is spread by eating or drinking food or water contaminated with the bacteria. Understood? It is an enteric pathogen. It can also be spread by direct contact with the feces from an infected person. And Shigella is a contagious infection. Okay. See, this is a photomicrograph of Shigella species in stool specimen. Got it? Now let's see its cultural characteristic. They are aerobes and facultative anaerobes. They grow best at 37 degrees centigrade. They require pH 7.4 on ordinary medium. Okay. Colonies are small, measuring about 1 to 3 into 0.5 micrometer, or you can say about 2 mm in diameter. Circular, convex, smooth and transparent. Okay. On McConkey's agar and DCA, deoxycholate citrate agar medium, they are pale or colorless except Shigella soni, which is late lactose fermenter. In Shigella salmonella agar, SS agar, the colonies are colorless. On XLD, xylose, lysine, deoxycholate agar. See, these are different mediums on which Shigella is grown, and how the colonies are formed, what's the changes, what sugars they are fermenting, everything is studied. Okay, so on XLD, Shigella produce pink colonies without black centers. Every medium has its own property. Understood? Here you can see the images. This is SS agar image. Salmonella, Shigella agar. This is on McConkey and this is nutrient agar. First is McConkey, second is nutrient. Okay. Next, biochemical characters. Now, carbohydrate utilization. Most strains utilize sugar to produce acid but not gas. Through some strain, Shigella flexneri and Shigella boydii form gases. Glucose is fermented by almost all strains. Lactose is not fermented within 24 hours. Means there are late lactose fermenter, as I told you in this. Yeah, Shigella soni. It is a late lactose fermenter. Okay. However, Shigella soni and some strains of Shigella dysentery produce acid from lactose after prolonged incubation. Understood? Mannitol fermentation is important characteristics. This differentiated group A strain which do not ferment mannitol, and group B, C, D most strains of which they ferment it. Understood? Dulcitol is not fermented by most Shigella. Sucrose is not fermented except Shigella soni and some strains of Shigella flexneri. Adonitol, inositol are also not fermented. Xylose is not fermented except mannitol. It's negative biotype of Shigella flexneri. Understood? C. Methyl red test positive, VP test negative, reduce nitrate to nitrite, catalase positive, indole negative, citrate negative. So the IMVIC will be negative, positive, negative, negative. H2S negative, urea is negative, casein growth is negative, gelatin not liquefied. Decarboxylation test. Group A, B, C failed to decarboxylate lysine and ornithine, and Shigella soni decarboxylate ornithine but not lysine. Understood? These are the biochemical characters. Now, antigenic structure of Shigella. Shigella are differentiated by their O antigens into serotype. These are classified into 4 structures or subgroups in combination with biochemical and serological characters. Group A, Shigella dysentery. These are mannitol non-fermenting, consist of 10 serotypes. Type 1 forms a toxin. 3 types of toxic activity have been demonstrated in Shigella culture filtrates. Which are the 3 types? Neurotoxicity, enterotoxicity, cytotoxicity. Understood? Now group B, Shigella flexneri. Now this name flexneri was named after Flexner, who first time described first of the mannitol fermenting Shigella from Philippines in 1900. Okay. Now based on type specific and group specific antigen, they have classified into 6 serotypes and several subtypes. Okay. So when I'll provide you the notes, you'll get it. Shigella boydii, group C, consists of dysentery bacilli that resemble Shigella flexneri biochemically but not antigenically. Understood? After Boyd, who first described this strain in India, 1931, Shigella boydii isolated least frequently. Okay, and 15 serotypes have been identified. Shigella soni, group D, isolated first time by Sone, 1915 in Germany. Ferment lactose and sucrose late. It is indole negative and causes mildest form of bacillary dysentery. Understood? In the next lecture, we will see its rest topics like pathogenicity, pathogenesis, symptoms, laboratory diagnosis. Okay? Any doubts? You can ask me. Thank you.